Edward Nikolayevich Trifonov, Hebrew, Russian, Abiogenic, March 31, 1937, is a Russian-born Israeli molecular biophysicist and a founder of Israeli bioinformatics. In his research, he specializes in the recognition of weak signal patterns in biological sequences and is known for his unorthodox scientific methods. He discovered the 3BP and 10BP periodicity in the DNA sequences, as well as the rules determining the curvature of DNA molecules and their bending within nucleosomes. Trifonov unveiled multiple novel codes in biological sequences and the modular structure of proteins. He proposed an abiogenic theory of the origin of life and molecular evolution from single nucleotides and amino acids to present-day DNA and protein sequences. Biography. Trifonov was born in Leningrad, now St. Petersburg, USSR in 1937. He was raised by his mother, Reba, and his stepfather, Nikolai Nikolayevich Trifonov. In his school years, he became interested in medicine and physics. As a result, he went to study biophysics in Moscow. He started his scientific career in the USSR. In 1976, he made Aliyah, immigrated as a Jew, to Israel. His role model is Gregor Mendel. Education and scientific career. Trifonov graduated in biophysics from the Moscow Institute of Physics and Technology in 1961 and earned his PhD degree in molecular biophysics there in 1970. He worked as a researcher at the Moscow Physico-Technical Institute from 1961 to 1964. Then he moved to the biological department at the I.V. Kurchatov Institute of Atomic Energy in Moscow, staying there until 1975. After his immigration to Israel, he joined the Department of Polymer Research at the Weizmann Institute of Science as an associate professor. He worked there from 1976 to 1991 before moving to the Department of Structural Biology as a full professor in 1992. He was appointed Professor Emeritus in 2003. During that time, he was also head of the Center for Genome Structure and Evolution at the Institute of Molecular Sciences in Palo Alto, California, 1992 to 1995. Trifonov has been a head of the Genome Diversity Center at the Institute of Evolution at the University of Haifa in Israel since 2002, and a professor at Masaryk University in Brno, Czech Republic since 2007. Membership of Learned Societies. USSR Biochemical Society. 1970. The Israel National Committee for CODATA. 1987. International Society of Molecular Evolution. 1993. International Society of Gene Therapy and Molecular Biology. 1997. Editorial and Advisory Boards. Editor. Microbiology and Biochemistry sections of Russian Biological Abstracts. 1970 to 1975. Editor. Journal of Biomolecular Structure and Dynamics. 1988 to 1995. Editorial Board and Associate Editor. Journal of Molecular Evolution. 1993 to 2004. Academic Council of the College of Judea and Samaria. Kedrumi Mariel, West Bank. 1994 to 1999. Editorial Board of Gene Therapy and Molecular Biology. Since 1997. Editorial Board of OMICS. Journal of Integrative Biology. Since 2006. Editorial Advisory Board. Biomolecular Structure and Dynamics. Since 2010. Research. At the beginning of his scientific career, Trifonov studied characteristics of the DNA with biophysical methods. After his relocation to Israel in 1976, he switched over to bioinformatics. and established the first research group for the discipline in the country. He is known for his innovative insights into the world of biological sequences. Research areas equals periodicity in biological sequences equals. Trifonov pioneered the application of digital signal processing techniques to biological sequences. In 1980, he and Joel Sussman used autocorrelation to analyze chromatin DNA sequences. They were the first to discover two periodical patterns in the DNA sequences, namely 3BP and 10 to 11 BP. 10.4. Periodicity equals chromatin structure equals. Since the beginning of his Israeli scientific period, Trifonov has been studying the chromatin structure, investigating how certain segments of the DNA are packed inside the cells in protein DNA complexes, called nucleosomes. In a nucleosome, the DNA winds around the histone protein component. The principle of this winding, and thus the rules determining nucleosome positions, was not known at the beginning of the 1980s, although multiple models had been suggested. These included the Hinge Model. The DNA molecule was assumed to be a rigid rod-like structure interrupted by sharp kinks, up to 90 degrees, with the straight segments being a multiple of 10 BP long. The isotropic model. The DNA molecule is bent smoothly along its length, with the same angle between every two base pairs. The mini kinks model. Similar to the Hinge model, but with smoother kinks every 5 BP. Trifonov supported the concept of smooth bending of the DNA. However, he proposed that angles between the base pairs are not equal, but their size depends on the particular neighboring base pairs, thus introducing an anisotropic, or wedge model. This model was based on the work of Trifonov and Joel Sussman, who had shown in 1980 that some of the D-nucleotides, nucleotide dimers, are frequently placed in regular, periodical distances from each other in the chromatin DNA. This was a breakthrough discovery initiating a search for sequence patterns in the chromatin DNA. They had also pointed out that those D-nucleotides repeated with the same period as the estimated pitch, the length of one DNA helix repeat, of the chromatin DNA, 10.4 BP. Thus in his wedge model, Trifonov supposed that each combination of neighboring base pairs form a certain angle, specific for these base pairs. He called this feature curvature. Moreover, he suggested that in addition to curvature, each base pair's step could be deformed to different extent, being bound to the histone octamer, and he called it bending. These two features of DNA present in the nucleosomes, curvature and bending, have been now considered major factors playing a role in the nucleosome positioning. Periodicity of other D-nucleotides were confirmed later by Alexander Bolshoi and co-workers. Finally, an ideal sequence of the nucleosomal DNA was derived in 2009 by Gabdank, Barash and Trifonov. The proposed sequence CGRAAATTTYCG, R standing for a purine, A G, Y for a pyrimidine, C a T, expresses the preferential order of the D-nucleotides in the sequence of the nucleosomal DNA. However, these inferences are disputed by some scientists. Another question closely related to the chromatin structure which Trifonov pursued to answer was the length of the DNA helical repeat turn within nucleosomes. It is known that in free DNA, i.e., DNA which is not part of a nucleosome, the DNA helix twists 360 degrees per approximately 10.5 BP. In 1979, Trifonov and Thomas Betkin estimated the length of a nucleosomal DNA repeat to be 10.33 to 10.4 BP. This value was finally confirmed and refined to 10.4 BP with crystallographic analysis in 2006. equals multiple genetic codes equals. Trifonov advocates the notion that biological sequences bear many codes contrary to the generally recognized one genetic code, coding amino acids order. He was also the first to demonstrate that there are multiple codes present in the DNA. He points out that even so-called non-coding DNA has a function, i.e., contains codes, although different from the triplet code. Trifonov recognizes specific codes in the DNA, RNA and proteins. In DNA sequences, chromatin code. Trifonov 1980 is a set of rules responsible for positioning of the nucleosomes in RNA sequences. RNA to protein translation code. Triplet code. Every triplet in the RNA sequence is translated to a specific amino acid. Splicing code is a code responsible for RNA splicing. Still poorly identified. Framing code. Trifonov 1987. The consensus sequence of the mRNA is GCU-N, which is complementary to XXC-N in the ribosomes. It maintains the correct reading frame during mRNA translation. Translation pausing code. Mac-Howell and Trifonov 2002. Clusters of rare codons are placed in the distance of 150 bp from each other. The translation time of these codons is longer than of their synonymous counterparts, which slows down the translation process and thus provides time for the fresh synthesized segment of a protein to fold properly. In protein sequences, protein folding code. Berezovsky, Grossberg and Trifonov 2000. Proteins are composed of modules. The newly synthesized protein is folded a module by module, not as a whole. Fast adaptation codes. Trifonov 1989 are present in all three types of biological sequences. They are represented by tandem repeats A, B, M, N, N. The number of repetitions, N, can change in the cell genome as a response to stress, which may or may not help the cell to adapt to the environmental pressure. Codes of evolutionary past. Binary code. Trifonov 2006. The first ancient codons were GGC and GCC, from which the other codons have been derived by series of point mutations. Nowadays, we can see it in modern genes as mini-genes, containing a purine at the middle position in the codons, alternating with segments having a pyrimidine in the middle. Nucleotides. Genome segmentation code. Kolker and Trifonov 1995. Methionines tend to occur every 400 bits per second in the modern DNA sequences as a result of fusion of ancient independent sequences. The codes can overlap each other so that up to 4 different codes can be identified in one DNA sequence, specifically a sequence involved in a nucleosome. According to Trifonov, other codes are yet to be discovered. Modular structure of proteins equals. Trifonov's concept of protein modules tries to address the questions of proteins' evolution and protein folding. In 2000, Trifonov with Berezovsky and Grossberg studied protein sequences and tried to identify simple sequential elements in proteins. They postulated that structurally diverse closed loops of 25 to 30 amino acid residues are universal building blocks of protein folds. They speculated that at the beginning of the evolution, there were short polypeptide chains which later formed these closed loops. They supposed that the loop's structure provided more stability to the sequence and thus was favored in the evolution. Modern proteins are probably a group of closed loops fused together to trace the evolution of sequences. Trifonov and Zachariah Frenkel introduced a concept of protein sequence space based on the protein modules. It is a network arrangement of sequence fragments of the length of 20 amino acids obtained from a collection of fully sequenced genomes. Each fragment is represented as a node. Two fragments with certain level of similarity to each other are connected with an edge. This approach should make it possible to determine function of uncharacterized proteins. Protein modularity could also give an answer to the Leavenworth's paradox, i.e., the question how a protein sequence can fold in a very short time, equals molecular evolution and the origin of life equals. In 1996, Thomas Bettkon, a German geneticist, noticed that most of the triplet expansion diseases can be attributed only to two triplets, GCU and GCC, the rest being their permutations or complementary counterparts. He discussed this finding with Trifonov, his friend and colleague. Trifonov had earlier discovered GCU N to be a hidden mRNA consensus sequence. Thus, the combination of these two facts led them to the idea that the GCU N could reflect a pattern of ancient mRNA sequences. The first triplets, since GCU and GCC appeared to be the most expandable, or the most aggressive, triplets, Trifonov and Bettkon inferred that they could be the first two codons. Their ability to expand rapidly comparing to other triplets would provide them with evolutionary advantage. Single-point mutations of these two would give rise to 14 other triplets. Consensus temporal order of amino acids. Having the suspected first two triplets, they pondered which amino acids appeared the first, or more generally in which order all the proteinogenic amino acids emerged. To address this question, they resorted to three, according to them the most natural, hypotheses. The earliest amino acids were chemically the simplest. They would be present among the products of the Miller-Urey experiment. They would be associated with the older one of the two known classes of amino acid tRNA synthetases. Later on, Trifonov collected even 101 criteria for the amino acids order. Each criterion could be represented as a vector of length 20. For 20 basic amino acids, Trifonov averaged over them and got the proposed temporal order of the amino acids emergence, glycine and alanine being the first two ones. Results and predictions. Trifonov elaborated these concepts further and proposed these notions. Evolutionary table of the triplet code. Glycine content of a protein can be used as a measure of the respective protein age. Glycine clock. Proteins are composed of short oligopeptides derived from ancient sequences being either oligoalanines or oligoglycines. Thus two alphabets. These two alphabets, distinguished by the type of nucleotide in the middle positions within triplets, purines or pyrimidines, provide us with a binary code, which can be used for more accurate analyses of proteins. Relatedness. Definition of life. A part of Trifonov's work on the molecular evolution is his aim to find a concise definition of life. He collected 123 definitions by other authors. Instead of dealing with logical or philosophical arguments, he analyzed the vocabulary of the present definitions. By an approach close to the principal component analysis, he derived a consensus definition. Life is self-reproduction with variations. This work gained multiple critical comments. Research techniques and approaches. Linguistic sequence complexity Linguistic sequence complexity, LC, is a measure introduced by Trifonov in 1990. It is used for analyses and characterization of biological sequences. LC of a sequence is defined as richness of its vocabulary, i.e., how many different substrings of certain length are present in the sequence. Terminology DNA curvature versus DNA bending Trifonov strictly differentiates between two notions. Curvature, a property of free DNA which has curvilinear shape due to slight differences in the angles between neighboring base pairs. Bending, a deformation of DNA as a result of binding to proteins, e.g., to the histone octamer. Both of these features are directed by the particular DNA sequence. Multiple genetic codes While the scientific community recognize one genetic code, Trifonov promotes the idea of multiple genetic codes. He adverts to recurring events of a discovery of yet another, the second, genetic code. Honors. Kurchatov Prize for Young Scientists. 1969. Kurchatov Prize for Basic Research. 1971. Kleeman Professor of Molecular Biophysics. 1982 to 2002. Adjunct Professor of Lomonosov, Moscow State University. 1999. The Stanislaw Ulam Memorial Lecture at the 2003 RECMB meeting. Berlin. 2003. Mendel Lecture. BRNO. Abbey of St. Thomas. 2004. Distinguished Membership Award of ISBCB. 2008. Distinguished Citizen Fellow University of Indiana South Bend, Indiana, USA. 2009. Citations. Trifonov, Edward N. Lecturer. 2010. Second, third, fourth genetic codes. One spectacular case of code crowding. Prague, Czech Republic. Archived from the original MP3 on the 10th of October 2010. Retrieved the 26th of March 2012.